Hello again. This is video number 2 of 4 in the week 2 review, unit review. The first video was rather lengthy. I hope you were able to make it through. We talked about the brain and the nervous system. This video is going to be much shorter. We're going to talk about sensation and perception. And so first we want to be able to distinguish between those two terms, sensation and perception. Sensation is sometimes referred to as bottom-up processing. And what that means is sensation occurs anytime a receptor cell in one of your 5 senses is activated. And because the brain is at the top of your body, all of your senses are below, and so when something goes from your senses to the brain, another way of describing that is bottom-up processing. Remember in the last video we talked about the afferent pathway, the sensory pathway, right? Sensory neurons. When information activates your skin, your nose, your ears, your mouth, that information goes through the spinal cord to the brain from the bottom up. The term for this is sensation. But as you learned in my class, I hope this does not finally determine what you see, what you hear, and what you sense. What you finally see and hear is going to be determined by how your brain perceives it. This is why people are able to see things that aren't there, and hear and feel and smell and taste things that aren't there. Because hearing and smelling and tasting is not created by your mouth and your hands and your nose, but rather in your brain. This is called top-down processing, also known as perception. Generally speaking, we all have a pretty common idea of the way things smell and taste and look, but there's no way to know for sure if the way that I perceive things is the same as the way you perceive them, and we definitely can't know for sure if the way that you perceive them is the same as your dog or your cat perceives them. This is what's happening in their brains. Perception, top-down processing. One of the most important words in this chapter, in this unit, is transduction, right? Your brain can't take the light that goes in your eyes. Light goes in your eyes and it can't go into your brain. So at some point in your eye, light has to be changed into a type of energy that your brain can process. Neural energy. Remember unit 1, the previous video. Neural energy. Things that neurons, the information that travels on neurons, neural information. Your eyes change light into neural energy. This is transduction. Your ears change vibrations into neural energy. Your mouth, your nose change chemicals into neural energy. All of these are transduction. Transduction is the changing of physical energy, light, sound, chemicals into neural energy. So how much light does it take for us to see it? How much sound does it take for us to hear it? How much pressure does it take on our skin for us to feel it? The answer is, as soon as we can detect a stimulus, about half the time, we say that that stimulus has reached our absolute threshold. So again, the absolute threshold is the amount of a stimulus, light, sound, pressure, the minimum amount of a stimulus that is required for us to perceive it at least half the time. Now, if there's a light on and it gets very slightly brighter, will you notice? If there's music playing and it gets very slightly softer, will you notice the change? And the answer is If the change is enough for you to notice it at least half the time, then we say that the change, not the amount of stimulus this time, but the change in the stimulus has reached your difference threshold, right? Also known as the just noticeable difference. So what's the difference between the absolute threshold and the difference threshold? The absolute threshold is the total amount of a stimulus, light, sound, pressure, that is necessary for us to notice it at all. The just noticeable difference, or the difference threshold, is the change that has to be made in the intensity of a stimulus for us to notice that there has been a change. Now, sometimes we stop noticing things. There are two reasons why we stop noticing things. One of them, the most simple, which is not listed here on your review, so just listen, is habituation. You remember habituation. It starts with the word habit, right? Habituation means that there's a sound or some stimulus and it's been there so long and it's so unimportant that you no longer notice it. But you can if your attention is brought to it, right? There's a very slight buzzing in the background being made by some electrical device. And you can only hear it when you pay attention. When you stop paying attention and you're working on something else, it disappears. This is called habituation. But sometimes the ability to notice a stimulus disappears, and no matter how hard you try, you can't make it reappear. For example, you jump into a cold swimming pool, and after 5 or 10 minutes, you can't again notice how cold it was when you went in, even though the temperature of the water has not really changed. Why? Because the receptor cells on your skin have become desensitized. That stimulus has been present on the receptor cells of your skin so long that they're no longer receiving the information. This is why you don't smell your own BO. Your own BO has lingered in your nostrils for so long that the receptor cells in your nostrils are no longer activated by those particular molecules. But trust me, you smell, whether you think you do or not. This is called sensory adaptation. Habituation is a matter of putting your attention on something or not. This is called selective attention. At this moment, I hope you're choosing to put your attention on me. And if you do that, there are going to be stimuli in the background of your home that you will no longer notice. Habituation, due to selective attention, focusing your attention on one thing and blocking out others. Sensory adaptation happens on the sensory level. Your receptor cells are no longer activated by a particular stimulus. Let's talk about vision. If you don't mind, please flip your review sheet over. I really think there are some important terms for vision that you need to be aware of, other than the three that I see listed here. Let's start with light entering your eye. The first structure of your eye is the cornea. It's a clear outer covering. It holds the shape of your eye, and it slightly bends the light as it enters your eye. Then light enters through the dark black middle of your eye called the pupil. And most of you already know that your pupils dilate, that is, get larger in darkness to take in as much light as possible, and they contract, that is, get smaller when you're surrounded by a lot of light to block out excess light. But this doesn't happen automatically without a muscle that opens and closes it. And it turns out the colorful ring around your pupil, called the iris, is that muscle. The colorful ring around the pupil is the iris, and it is a muscle that opens, dilates, and closes, contracts the pupil to let in more and less light. Behind the pupil, inside the eye, next is the lens. The lens, like the lens in these glasses, is thick, and it changes shape. It changes shape to focus on things. You notice old people, when they're in a restaurant looking at a menu, they stretch their face. And they do that to make the lens thinner so they can focus on things that are close. Or we squint sometimes to see things that are far away. And that is, we are squishing, making the lens thicker to focus far away. When you're young and healthy, the lens does this normally by itself, and this is called visual accommodation. The light goes through the pupil, through the lens, and then the fluid-filled center of your eye all the way to the back wall of your eye called the retina. When the light travels through the fluid in the middle of your eye, like any other time that light goes through liquid, it inverts the image. It turns it upside down. So the image that you're looking at when it lands on your retina is upside down. So why don't we see the world upside down? Because your brain knows that it's not supposed to be upside down. And so when the upside down image gets to your brain, you never see that. Your brain, before you get a chance to see what's there, is inverting it. You remember some of you wore the inversion goggles that I have in my room. If you wear those goggles long enough, your brain will stop inverting the image because it knows that the world's not upside down. So the retina is the back wall of your eye where the light is projected. The middle of the retina is called the fovea, F-O-V-E-A, fovea. And it's in this fovea where you have the rods and the cones. Now going back to the front of your video, front of your review sheet, if you want to, the rods and the cones are sometimes called photoreceptor cells. This is where transduction occurs, right? Light is changed into neural energy, transduction. The rods help you process, transduct dim light. The cones help you process color. While we're on the subject of color, there are two theories of color vision that you need to be aware of. The older theory is called as in three, trichromatic. This theory suggests that three types of cones, like the three primary colors, combine to create every possible hue. A more recent theory is the opponent process theory. And that's the idea that cones are in pairs, blues with yellows, reds with greens. And through the process of these cones being activated by certain light frequencies, it helps your brain to image all the different possible hues. So opponent process theory of color suggests that you have two kinds of cones, blue, yellow, and red, green cones. Now, all this information, after it is transducted into neural energy, has to travel on neurons to the brain. And what kind of neurons take information from the senses to the brain? That's right, class, good. Afferent, sensory neurons. And do they ever travel alone? No, of course not. They can't travel alone. They're too thin and fragile. They travel in bundles coded in myelin. So this particular bundle of nerves that travels from the back of your eye to the back of your brain is called the optic nerve, a bundle of neurons coded in myelin, taking this transducted visual information to the back of your brain. Each eye has a left and right field of vision. and everything in your right field of vision goes to the left occipital lobe and vice versa. Each eye has its own right and left field of vision that travels to the opposite occipital lobe, visual cortex, via the optic nerve. Moving on to hearing, right? You have a nice diagram here, so I'm going to go through it with you. The outer ear, starting with the pinna, this part that many people get pierced. This is number 1 on your diagram. Number 1 is the pinna, P-I-N-N-A, and its function is to funnel vibrations into the middle of your ear. Number 2 is the ear canal. Be careful what you stick in there. Remember I told you about my friend Cecil Gaffney? Went too far with the Q-tip. It was awful. The auditory canal funnels vibrations to the eardrum, which is number 3, the eardrum. When the vibrations hit the eardrum, it vibrates, just like hitting a regular drum causes the skin on the drum, the membrane, to vibrate. 1, 2, 3. After number 3, this diagram gets pretty funky, and I appreciate with that. I apologize for that. So let's talk about the middle ear, right? The middle ear consists of 3 bones, the hammer, the anvil, and the stirrup. And I would say on this diagram, that would be 10, 9, and 4. Don't ask me why. No idea. But 10, 9, and 4 would be the hammer, 10, 9 the anvil, and 4 the stirrup. I didn't make this diagram. I borrowed it because I was too lazy to make my own, but I didn't make it. Now, the purpose of the middle ear is conduction, that is moving sound from the outer to the inner ear. The most common kind of hearing loss for old people is conductive hearing loss. These 3 bones, the hammer, the anvil, and the stirrup, are banging together, rattling all the time, and like any other hard structure, they tend to wear out. Conductive hearing loss can easily be fixed by simply putting in a hearing aid, which I've been told I should do pretty soon, sooner than later. All right, the inner ear, the cochlea, number 7, it looks like a snail. You see number 7? Looks like a snail. The snail-like structure in the inner ear is the cochlea. And the semicircular canals, number 8. We're going to come back to the semicircular canals in a minute. But number 8 is the semicircular canals. The basilar membrane is number 5. Basilar membrane. Remember my hair cell dance, right? A membrane is simply skin. And think about the skin on your body. Even you ladies, if you look closely enough, you see little hairs. It's very typical for skin to be covered in hairs. Inside the inner ear, floating in fluid, are these hairs, and when they move, this is what creates transduction. Vibration is changed into neural energy, right? The loud noises stimulate the hair cells, which creates the neural energy that our brain processes as sound. Different frequencies, high frequencies and low frequencies, are processed at different places on the membrane. This is called place theory. Place theory is the idea that where in the membrane a sound is activated or transducted determines the frequency. That place then is determined by the wavelength. Long waves, low frequency. Short waves, high frequency. So the length of the wave determines where in the inner ear it's activated, which then determines the frequency of the sound that we hear. Did I cover them all? Oh, the auditory nerve, number 6. What is the auditory nerve? What is any nerve? A bundle of neurons covered in myelin, right? Taking information to the brain. What's the name of that middle section? What's that structure in the middle of the brain that it has to pass through all senses except smell? That's right, the thalamus. The thalamus on the way to the auditory cortex. On the sides of the brain, the temporal lobes contain the auditory cortex. Finally, when it comes to how we perceive things, through evolution, the human mind, the human brain has developed some common ways that we interpret our environment. These ways were discovered by German psychologists who used the term Gestalt. Gestalt means the whole. When you look at another person, you see a single person. You don't see millions of little parts, or even hundreds, or even dozens, or even a few. You see one person. The reason for that is because over time your brain has learned that that's the best way in terms of survival to see things in meaningful wholes, not in individual parts. Therefore, the way that your brain interprets different parts into meaningful wholes is sometimes called Gestalt principles. We talked about something called perceptual constancy. If you know that an apple is red, it will look red even in total darkness to you, because your brain will help you or cause you to see red even though there's no light being reflected off of it, because your brain knows that in fact it is red. If your brain knows that a door is a rectangle, it doesn't matter at what angle you look at it, it will still be interpreted as a rectangle. Figure ground is the idea that what you focus on through selective attention is figure, and everything else is ground. The famous vase, that is a vase if you look at it one way, or two faces looking at each other if you look at it a different way, is an example of figure ground. When things are similar, we tend to group them together. That's similarity. When things are close together, we tend to assume they belong together. That's proximity. When a circle has gaps in it, our brain tends to see it as a circle anyway. That's called closure. And when a line is broken, we tend to see it as a continuous line because of something called continuity. All of these, similarity, proximity, closure, continuity, they're all the way that your brain tries to make meaningful sense out of bits and pieces of the environment. And the term for this is called Gestalt. How do we know how far away something is? There are two kinds of cues. There are two kinds of depth cues. And by the way that depth perception is tested, we'll get to that in a minute. Let's see. So the first kind of cues, the first set of cues are called binocular. Bi meaning two, ocular, eyes, two eyes. As my eyes get closer and closer to the screen, you notice that they have to roll inward, okay? When they roll inward, that's called convergence. When my eyes roll inward, the muscles that move them inward communicate with my brain, telling my brain what? That thing is getting closer. When I back up away from the screen, my eyes relax and widen. My brain interprets this as things getting farther away. Also, remember that each eye has its own retina. And the image in my right retina is not exactly the same as the image in my left retina. And the closer I get to the screen, the more different those two images are. The farther I get from the screen, my left and my right eye have two very, very, very similar images. But the closer I get to the screen, my right and my left eye have two quite different images. Your brain is so smart, so highly sophisticated, that it can use the disparity, that is the difference in those two images, to help calculate or guesstimate how far something is. Convergence and retinal disparity, those are binocular cues that your brain uses to interpret how far away things are. The other cues you learned in art class. Remember art class? You learned how to make it look like somebody was standing behind somebody else by drawing only part of the person in the back as if they were being blocked by the person in the front, when in reality they're both on the same piece of paper. In real life, if somebody is standing behind someone else, you can't see them or not, you can't see their whole body. This is called interposition. And your brain knows, or thinks it knows, that if it can't see all of one person, that it must be because the other person is closer. Relative size. We know that all human beings are pretty much similar, somewhere between 5 feet and 6.5 feet. And so therefore, if somebody appears half of the size of somebody else in your retina, they must be farther away. If you're drawing in art class, you make somebody smaller if you want to give the illusion that they're farther away. You make them larger if you want to give the illusion that they're closer. Relative size. The same thing with clarity. When you're drawing in art class, if you want to give the illusion that something's far away, you can fuzz it up. You can distort it so that it can't be clearly seen. Whereas if you want to give the illusion that something's very close, you can give it a lot of clarity. Clarity, relative size, interposition, all of these are monocular cues. That means you can do them with one eye. You don't need two eyes for depth perception because of monocular cues. How do psychologists study when babies develop depth perception, or whether or not certain animals possess depth perception? And the answer is a very famous or well-known experiment called the visual cliff. Remember the tabletop that had a red and white checkerboard top on one half and then a glass top on the second half with a red and white checkerboard floor below it? The mommy would stand away from the baby on the opposite end and call for the baby to come. If the baby crawled over without a second thought, one can assume that they lack depth perception because it didn't occur to them that they could fall off the cliff. But if the baby stops and cries and calls out to mom and won't go over the glass edge, even though there's glass there, we can interpret that as having depth perception. This is called the visual cliff. Finally, we will conclude this section with the last of the senses, right? The last of the senses are called somaesthetic. That means body feeling senses, right? Your skin can feel. Just like your mouth has 5 different kinds of taste buds, your skin has 4 different kinds of receptors for temperature, for pain, for pressure, and for touch. This is called the skin senses. The skin senses are only part of the somaesthetic senses. The next part of the somaesthetic senses after skin senses is your vestibular sense. The vestibular sense is your sense of balance that's created by item number 8 in your ear diagram. Go back to your diagram and look at number 8. Do you see those semicircular canals, those tubes? When I lean one way or the other, the fluid in those semicircular canals is going to stay level, just like when I lean a bottle of water one way or the other. And where the fluid touches my ear when I lean automatically tells the rest of my body without thinking how to keep from falling. When you are knocked from one side or the other, your body does involuntary things automatically to keep you from falling. And these things are being commanded, these commands are being given by your vestibular sense. Finally, you have your kinesthetic sense. I'm looking at the screen right now, but I can tell you where my feet are. You know why? Because I can feel them down there. So no matter where I am or what I'm looking at or what I'm thinking about, I always have either a conscious or an unconscious awareness of where my body parts are at any given time. I might not be thinking about it. When I think about it, I can feel them. But even when I'm not thinking about it, my brain is giving me a message. My brain is receiving a message from my feet as to their location. This keeps me from bumping into things when I walk around. Elite athletes, dancers, musicians have a very strong kinesthetic sense, right? And what part of the lower brain do you think is very closely associated with kinesthetic sense? That's right, the cerebellum. The cerebellum. Remember, the athlete's brain. The more you practice your sport, the more you practice a movement, the more involuntary it becomes. Your awareness of where your arms and legs are when you're dancing, when you're playing your sport, becomes second nature. This is your kinesthetic sense. All right, so I hope that you found this second video very helpful. You should be just about done with your review sheet now, except for the small portions that can be completed by watching the crash course videos. Peace.